Genetic engineering is a modification of genetic information of living organisms by manipulation of DNA, like by adding, removing, or repairing part of genetic material and changing the phenotype of organism. Paul is considered as a father of genetic engineering. It is one of the modern methods of biotechnology and is also known as gene manipulation or recombinant DNA technology. It involves three components: donor gene, recipient gene, and vector. How a genetic engineering works? Yes, it involves four steps. First is isolation of DNA of interest. Here, gene is obtained from donor organism. Second step is to cut DNA with restriction endonucleases. Restriction endonucleases are molecular scissors or genetic scissors. These are used to cut the genes. These enzymes cut at the restriction site of gene. Enzyme ligase is used to join genes in specific area. That's why it is called molecular glue. In next step, target gene is combined with vector DNA. Vectors are vehicles to carry DNA. When target gene is incorporated with vector DNA, it is known as recombinant DNA. Plasmids are widely used vectors. In the final stage, recombinant molecule is introduced into the host cell. Genetic engineering has wide variety of scopes in medicine, agriculture, research, etc. Genetic engineering is used in the field of medicine to produce different types of drugs, growth hormones, vaccines, monoclonal antibodies, etc. Genetically engineered insulin is the first application of RNA technology. In agriculture, genetic engineering can be used to produce high-yielding varieties of crops, disease-resistant varieties, and increase the production of milk and meat through genetically modified organisms. Genetic engineering advanced the research field through the introduction of genetically modified organisms, disease-resistant varieties, introduction of antibiotics, drugs, etc. It also advanced methods for conservation of resources, natural area, and also microbial art. Gene therapy. Gene therapy is a technique that modifies a person's gene to cure or treat the disease. Here, the undesired or abnormal gene that leads to disease can be eliminated or replaced with a healthy gene. It is useful to treat wide variety of genetic disorders. Human Genome Project added importance to gene therapy by identifying the complete set of human genome. Human Genome Project is the first international collaborative science project to determine complete sequence of human genome and genes in it. It was started in 1990 and successfully completed in 2003. DNA fingerprinting. DNA fingerprinting was developed by Alex Jeffries in 1984. It is the method of isolating and identifying variable elements within the base pair sequence of DNA. It has a wide variety of uses, like to solve parental disputes and to find out hereditary characters of living beings and find out missing individuals and criminals Another important feature of genetic engineering is the production of genetically modified crops like BT crops, flavor saver tomato, golden rice, and GM mustard. BT cotton and BT maize are the crops that are resistant to Bacillus thuringiensis, soil-dwelling bacteria that decreases the production of crops. Flavor saver tomato is a tomato resistant to rotting. It is the first commercially grown genetically modified crop. Golden rice is a GM crop with high content of vitamin A. Genetically modified animals are produced to fight against different types of diseases and to increase production of milk, meat, etc. Thus, genetic engineering paved importance in all fields of humankind. It has several advantages like the production of insulin, growth hormones, genetically modified animals, and increased productivity of food. Even though it has so many advantages, it also faces challenges. Most of the techniques affect the environment in a negative way. Altering the gene also alter microbial diversity that protect the environment. Some of the techniques may also cause health issues. It also leads to metal toxicity. In short, genetic engineering is advanced technology that have both impact and disadvantages to living things in the earth.